Mirror bacteria, a radical discovery that redefines life on Earth. The truth is AI. Mirror bacteria, synthetic organisms with reversed molecular structures, present a groundbreaking yet precarious avenue in the field of synthetic biology. While still a distant possibility, their creation could redefine biological principles as we know them. In this analytical deep dive, we will explore the science, implications, and ethical dilemmas surrounding these microorganisms that scientists warn may constitute a radical departure from known life. Imagine a world where life exists in a form that mirrors our molecular structures, not simply differing in complexity, but fundamentally distinct in design. These are mirror bacteria, synthetic life forms whose molecular chirality, handedness, is opposite to that found in nature. On Earth, all life conforms to a specific chirality, such as DNA's right-handed helices or amino acids' left-handed orientation. Mirror bacteria, however, defy this biological uniformity by mirroring these configurations. Chirality forms the cornerstone of biological processes. From enzyme interactions to the immune system's ability to detect pathogens, life relies on subtle molecular shapes. However, mirror bacteria would operate outside this paradigm. This raises profound questions. How would mirror organisms interact with natural ecosystems? And how might they affect the delicate balance of life? The concept of mirror bacteria is theoretical today, but the alarm bell has already been rung by a collaborative study involving 38 scientists from 9 different countries. These researchers explored the scientific feasibility and potential biosecurity risks of such organisms. Their consensus is clear. The emergence of such life forms could have catastrophic consequences, even though their realization may be decades away. This isn't merely a speculative idea. The science builds on proven principles of synthetic biology, the manipulation of cells and molecules to engineer life. Although creating mirror bacteria would require breakthroughs on the scale of the Human Genome Project, the possibility of achieving these milestones cannot be ignored. Developing mirror bacteria would involve overcoming significant technological hurdles. Reverse engineering life to this scale demands a blueprint akin to humanity's decoding of the genome, but exponentially more complex. Recent breakthroughs in synthetic biology, such as producing artificial DNA molecules, provide hope but reveal the vast undertaking ahead. While the process may require decades of effort and massive global collaboration, synthetic biology has proven its ability to produce innovation remarkably fast. The very possibility of achieving such technology invites both scientific excitement and calls for caution against unintended consequences. What happens when life forms fundamentally different from natural organisms enter the environment? Ecosystem disruption is one inevitable scenario. Existing biological systems have co-evolved based on the universally shared chirality. The introduction of mirror bacteria could shatter these balances. Perhaps even more alarming is the potential impact on human health. Our immune systems, designed to identify specific molecular shapes, could fail to recognize mirror bacteria as threats. Such organisms could circulate unnoticed, multiplying without restraint. A small outbreak might translate into widespread disruption, not just to humanity, but also to entire food chains and vital ecosystems. The intricacies of how mirror organisms behave remain an unanswered question, making their potential dangers all the more unsettling. Even with strict containment protocols, nature has a way of escaping control, raising concerns about looming biosecurity risks. As radical as they may sound, scenarios involving mirror bacteria ignite deep ethical dilemmas about humanity's relationship with creation. At what point does scientific curiosity outweigh the risks? Are we prepared to accept responsibility for letting the genie out of the bottle? Once released, organisms this transformative cannot be unmade. Their unique molecular makeup grants them the potential to evolve and adapt, spreading in ways we may not yet fully comprehend. Past lessons in underestimating biotechnological advancements serve as stark reminders of what happens when caution is overlooked. This ethical reflection shines a critical lens on science itself. Innovation in the absence of governance can have irreparable consequences. Mirror bacteria might start as a controlled experiment, but could quickly become an unstoppable evolutionary force. The challenges posed by mirror bacteria draw striking parallels to lessons learned during projects like the Human Genome Project. Initially viewed as an impossible venture, the sequencing of the human genome transformed what we could imagine achieving in biology. However, it also underscored the importance of robust ethical debates and oversight. Similarly, CRISPR-Cas9 gene editing made waves for its precision in altering genetic material. While its potentials in medicine and agriculture are exciting, its misuse remains a credible fear. In this progressive history of synthetic biology, mirror bacteria could serve as either the next remarkable milestone or the most dangerous misstep. Given its scale and potential risks, the development of mirror bacteria transcends borders. Effective oversight demands international cooperation, much like coordinated efforts on climate change or nuclear disarmament initiatives. The need for a global regulatory agency under the umbrella of the United Nations or a dedicated scientific body becomes apparent. Such an agency would ensure that mirror bacteria research follows stringent bioethical guidelines while maintaining transparency. The aim isn't to stifle innovation, but to prevent misuse and avert ecological catastrophe, a classic exercise in weighing progress against precaution. While biosecurity concerns dominate discussions, the public health implications of mirror bacteria are equally sobering. A pathogen that bypasses conventional immune defenses could wreak havoc before detection systems can adapt. On a large enough scale, this could overwhelm healthcare systems worldwide, especially in under-resourced regions ill-equipped to manage such crises. Moreover, does humanity have the right to experiment with creating such life when the potential for misuse looms large? The line between innovation and public endangerment may blur rapidly without global oversight. The creation of life in a form we've never encountered challenges the very notion of what it means to be alive. For scientists, it's an ethical tightrope, advancing human knowledge against the backdrop of potential disaster. Scientists are custodians of knowledge, meant to explore the universe's mysteries while anticipating its pitfalls. The question is no longer about whether mirror bacteria should exist, but how humanity manages the consequences of such discoveries, both intentional and accidental. Ethical frameworks must strike a delicate balance. They must advocate measured progress while heeding centuries-old lessons about the unintended consequences of scientific curiosity. Could we, as a global society, afford to make mistakes when the stakes are this high? Regulations surrounding synthetic biology remain nascent but integral to ensuring safe innovation. Establishing safeguards that mandate transparent research and mitigate worst-case scenarios supersedes national jurisdiction. It becomes a human imperative. Policy discussions on mirror bacteria should start well before the technology arrives. Prevention requires us to act, not react. Safeguarding the future means proactive collaboration between governments, the scientific community, and non-governmental organizations. No single entity can handle the risks posed by mirror bacteria, and therein lies the critical challenge. Despite its risks, research on mirror bacteria holds incredible potential for biotechnology and medicine. For instance, microorganisms with reverse chirality could lead to breakthrough treatments by interacting uniquely with human biochemistry. They could serve purposes we have yet to imagine, unlocking tools for long-term medical and environmental solutions. This dual-edged sword underscores an enduring question for synthetic biology. Can we responsibly explore potential benefits while minimizing existential dangers? The history of human innovation has often been marked by unintended consequences, but perhaps this time, with foresight, we can change our approach. Efforts to grasp the complexities and implications of mirror bacteria must include the broader scientific community. Informing and involving biologists, chemists, philosophers, and environmentalists is essential to ensure multidisciplinary solutions. Beyond academia lies the challenge of educating policymakers and the public. Demystifying such advanced concepts can garner informed dialogue, essential for decisions that shape the trajectory of synthetic biology. The creation of mirror bacteria isn't just a pivotal development in synthetic biology. It's humanity's first foray into a new form of life completely. Whether or not this represents evolution or hubris depends on cautious action moving forward. With the potential to disrupt the only ecosystems we've ever known, mirror bacteria demand our collective vigilance. They are no longer objects of fascination confined to ivory towers. They are urgent, real challenges transcending disciplines and borders. Scientific progress is inevitable, but let's remember, curiosity without caution risks irreversible consequences. Knowledge is a responsibility, and in the case of mirror bacteria, humanity must rise to this occasion with both humility and wisdom. Thank you for joining us on this fascinating exploration of mirror bacteria and the radical implications they have for our understanding of life. If you enjoyed this video, don't forget to hit the like button, share it with curious minds, and subscribe to The Truth is AI for more deep dives into the unknown. Turn on notifications so you don't miss our next video. Let us know your thoughts in the comments below. Could this discovery change how we view life in the universe? Until next time, keep questioning and keep exploring.